Question 6, we're looking at biotic and abiotic factors. So biotic are living factors, abiotic are non-living factors. So if we have a look here, so diseases, these are caused by pathogens which are living, so that is biotic. Herbivores, organisms that, or animals that eat plants, so they are alive, that's also biotic. Temperature, non-living, abiotic. Water, non-living, abiotic. Two students investigated the effect of light intensity on the distribution of small plants. The plants are growing under a tree in the park. As you move, their hypothesis is, as you move outwards from the tree, there will be more plant growth. Why would we think this is correct? So the tree would block light from reaching plants that grow under the tree. So which means less photosynthesis can take place. And this means less glucose or proteins which need glucose can be made for growth, so they don't grow as well. Next, student uses two pieces of equipment. Give the scientific name for each piece of equipment. So we have a square frame measuring 0.5 meters by 0.5 meters. The ones we've used in the past have been a meter by meter, but same idea, it's a quadrat. And an electronic device to measure light intensity, so it's something to meter light, it's going to be a light meter. We'll look at the method used. This is slightly different to one that we've seen before. They fixed one end of a tape measure at the base of a tree, they fixed the other end 11 meters away. At 0 meters they put the square frame, the quadrat, so essentially they've put one quadrat here. They identified all the plant species growing inside the frame. They estimate and record the percentage cover of each plant species, and measure the light intensity, and then put the square every 2 meters along until you get to 10 meters. So every 2 meters, sorry, will look like this. So they're placing it 1, 2, 3, 4, 5, 6 times. To work out the total area sampled, as it's asking us to do here, we'll need to do 6 times, because we've placed it twice, the area of the quadrat. So the area of the quadrat is 0.5 times 0.5. I think it's going to be 0.25, but always type it into the calculator. 0.25 meters squared. So 6 times 0.25 meters squared gives me, there we go, calculator, 1.5. So the total area sampled is 1.5 meters squared. So total area sampled 1.5 meters squared. Just a side note, this method of placing a tape or a rope across a habitat to measure the distribution of an organism is called a transect. So slightly different from just placing a quadrat randomly by throwing it with your eyes shut. That's going to give you population size rather than distribution. The whole investigation was done as quickly as possible on the same day. Well, if you'd have left it too long, or if you'd started it at midday and very slowly done it and finished it at 6 in the evening, the light intensity would not have remained constant, so that wouldn't give you a true reading. So light intensity would vary, and let's say due to time of day. or cloud cover. Give one away, the investigation could be improved. So they've only done one transect, one line in one direction of the tree. So you could repeat at different directions around the tree. So if I flip back over, so they've only done one direction. You could go over here, as many directions as you wanted really, until you're happy that the results are concordant. Next we have a table of results. So here we go, we've got the distance from the tree in metres, so 0, 2, 4, 6, 8, 10, and then you've got the percentage cover of the grass, your plantain, your daisy and your clover, and underneath here you've got a combined total percentage cover of all the plants. So as you can see, closer to the tree, that's low. Further away from the tree, it's 100%, the whole area is covered. And down here you can see that as you move away from the tree, the light intensity is increasing up to about 6 meters, beyond which it's at its maximum, it's over 200. Which plant species in table 7 will only grow at high light intensities? So we'll say these are high light intensities over here, anything over 175, and we'll go up our table and we'll find something that grows in this column, these columns, but not over here. And the daisy is the obvious one, because if you look at these low intensities, there's no daisies. So, daisy. Next, what conclusion can be made about the relationship between light intensity and the total percentage cover of plants? So, as our light intensity increased up to about 175, so did the total percentage cover of plants. Beyond 175, it remained constant at 100, so we need to phrase it that way. So, as light intensity increased, so did percentage or total percentage cover of plants. Up to a light intensity of 175 units, beyond which it stayed constant at 100%. So light intensity might not be the cause of this pattern of plant distribution. So yeah, plants definitely compete for light, but they also compete for water and nutrients, both of which might be absorbed by the tree. So water would be the obvious one. So the tree would block rain, or absorb through the roots. And as I mentioned previously, minerals or nutrients would also be fine, and again that's because the tree would be absorbing them through its roots, so it would outcompete the smaller plants trying to get hold of that.